It's 4 o'clock in the afternoon. Really sorry. I'm going to try and keep you awake. I'm hoping you guys will ask me questions. I've never given a talk like this before. You know, most times I'm giving talks where I understand what I'm talking about. Mostly. This time I have absolutely no clue, right? So I'm begging you guys, just join me, help me out, try to ask questions. These are tools of machine learning that I'm starting to use in trying to understand a scientific problem. And it turns out that I've gone and worked with people. Can I lower that? It's very loud. Okay. You know. How are you guys doing? Is it very loud or is it okay? No? Okay. Don't worry about it. So, you know, I'm starting to use the tools of machine learning, and that's been very complicated for me because I go talk to computer science people. They don't want to tell me what's behind. So I've been trying to figure things out. I started out as a user, and now I'm trying to go deeper in trying to understand. And so you will see a flavor of that, you know, that I'm going to start talking about as though it's a tool, and then try, if you guys want, we can go deeper in and ask those questions. I also designed it in a way, so I had my student, Tejas Shastri, put together Anaconda pages, which will run the sort of codes that I'm going to talk about. So for whatever reason, if you guys think it's exciting, you can look at it. And maybe, how many of you guys have done machine learning before? Wow, just one person. Cool. I'm not preaching to the converted. So the work I'm going to tell you about is funded by the Department of Energy and the Air Force Research Labs. I'm at Columbia. I'm in an engineering department. So I'm not a physicist. So this is sort of my model of what I'm thinking. I've got today's lecture set out, right? And I'm going to talk about that. I know the third lecture will be a classical talk. I'm going to talk about actually how we use these materials for gas separations and how we can start to design materials for gas separations. I'm going to tell you everything about the problem that I care about. The second one, I'm not sure what I'm going to talk about. I'll see how today goes and figure that out. One thing I was telling you, I already told you this the last half an hour or so. If my student comes online, he will help you with running code. If he doesn't, I will keep talking. Now, gas separation. I'm at a point in my life now where I want to do research that actually has value, right? So something that will actually change the human condition. And so I got into this problem, which is a very classic problem of sustainability. If I say gas separation membranes, how many of you guys have heard about that even? Few. Any reason why I should care? So right now, if you think about natural gas that's coming out, the US is the leader in what's called fracking technology, right? You go underground, you get gas. It's 70% methane, 30% CO2, right? And it's got something like H2S, it's got, you know, other compounds that can turn into acids. And so if you start pumping this gas, you've got toxic chemicals that you don't want. Also, CO2, you guys might have heard, is a pollutant that we don't really want to release into the atmosphere, right? So you want to get the CO2 out of the air so you can sequester that. So these are sort of the problems that we think about. And then, you know, in the US, we still burn hydrocarbons, right? So if you burn hydrocarbons in oxygen to produce power, you're going to get a mixture of CO2 and nitrogen. So you want to, again, take the CO2 out. So the focus here is on CO2, at least from what I'm thinking about. And I'm going to think about CO2 nitrogen separation, CO2 methane separation, right? These are the kind of problems that I care about. These are large volume separations. And right now there's a power plant in Texas, which is separating CO2 from nitrogen. And roughly a third of all the power that is being generated is used to affect the separation, okay? And so that means basically your power cost goes up by 50%. And it's that cost issue that makes these technology non-sustainable. Okay, everybody following along. Is this science enough for you guys, or does it sound like hocus pocus engineering stuff? I'm going to take you back and start talking science fairly quickly. All right. Now, if you think about these gases, right? So think about hydrogen, CO2. These are sort of canonical gases you want to separate. These are the standard diameters of these molecules. So if you want to separate CO2 from methane, that's a 3.3 versus 3.8 angstrom size difference. So if you're going to start doing things based on sizes, these are very hard, subtle size-based separation, okay? And CO2 and nitrogen, you can see again, is an even more difficult separation, okay? So this is the field that I want to play in. I want to start separating these mixtures, right? How do people do it? Yes. Who cares, man? Make a hole in the ground, stick it in there. Oh, I mean, there's many, so that's actually a very good question, right? So there's many thoughts on that. One is you're boring into the earth to get methane out, right? So you've got that reservoir in there, you put the CO2 back in there, and apparently that's a really good way to hold it, okay? The other thought which people have is to convert it to a carbonate, which is a solid, and then you bury it, right? The third idea is that you can actually take CO2, pump it to the bottom of the ocean, where it forms a liquid and it sits there. So there are ideas, but none of these at the moment is practically realizable. At the moment, the separation is so complicated, people haven't thought about what to do next. But it's a very important question to think of. Okay, so how do these people do separations right now? One idea is to do distillation, which is to say that CO2 has a boiling point that's very different from methane. You go in between the two boiling points and you can separate them out. One condenses, the other one comes. Okay. The problem is that you have to cool these things down to 190, 200 Kelvin to make those separations. That's very, very expensive. That's not feasible. So the idea people have used is to use what's called membrane separation. Okay. What do? You take a polymer, and I think Professor Kumar talked about that last week or so, right? Everybody knows what a polymer is, right? Or do I need to bore you with the details of what a polymer is? Please don't help me. So polymers come in many flavors. You can have polyethylene, polypropylene, polystyrene. There's like 10,000 known polymers. You take your favorite polymer, you form a membrane. On the upstream side, for example, here you put methane and CO2. And what you want to do is pick the membrane properly so that you get a high flux of CO2 on the other side, or the high flux of methane on the other side. Okay? So you want high flux, you want high purity. That's the figures of merit you're dealing with. How do you figure that out? How do you figure out the flux across the membrane? It follows straight from Fick's law. The gas dissolves into the membrane and then diffuses through. So the flux through the membrane, which is this diffusive transport, D is the diffusion constant. That's the concentration of gas in the membrane. X is the distance through the membrane. Very standard, right? If you assume some sort of Henry's law to define the concentration, then that reduces to this form, okay? I'm assuming basically a linear concentration gradient through the film. This then turns into the pressure drop times the solubility, that is the delta concentration, divided by the thickness, all right? So now if you think about the flux, this is the pressure drop across the membrane that you can fix. You know the upstream pressure, the downstream pressure, you know the thickness, and then the polymer itself contributes those two quantities, the diffusion constant and the solubility of a particular gas in that membrane. The higher that number is, the higher is the flux through that membrane. All right. Now, if you want to get high selectivity, it's the ratio of fluxes of the two quantities. You want that number to be large. So that means the ratio of permeabilities needs to be large. The permeability itself needs to be large. Those are the figures of merit you're trying to do. And what you're trying to figure out is which polymer should I use to simultaneously maximize permeability and selectivity. Okay. Now, everybody following this far, you're doing good, right? Yeah. Okay, just so long as, front row doesn't go to sleep, I'm doing good. And you will go to sleep eventually. So how does this mechanism work? I mean, people think, and there's no real understanding, it's like a sieve or a jali, right? So the polymer is sitting there, it's got some natural free volume pocket size, and depending on which polymer you pick, it has the right free volume size to let one gas go through and the other one not, okay? Now, if you have a jali that gets coarser and coarser, the flux goes up, but its selectivity drops because it no longer can distinguish between the two gases in question. You are squinting back there, are you following? Not you, the person in front of you. You, okay. So the idea is that it's a size-based separation people think, okay? And that is brought up to bear by these sorts of plots. This is a very engineering plot. I'm going to walk you through this because this is going to tell you everything that is a problem and what I'm trying to solve. So this is the permeability of CO2 in some crazy units. You don't care what that is. That's flux divided by pressure drop into the thickness. And that famously is called a bearer after the guy who came up with the unit. It's a nonsensical non-SI unit, okay? But the higher PCO2 is, the higher is the flux. This is a ratio of fluxes, that's a selectivity, right? Now you see here that data generally go that way. It tells you as I make the sieve coarser, permeability goes up, but the selectivity drops. Okay, so that correlation is beautifully there. And you can see here, there is a line here, or there is a line here below which all the data flows. That's called the upper bound correlation. Okay, now that's the data as it exists now. This is from 2010 or 2012 or whatever. You'll see many, many points. Those are, each one is a separate polymer construct. Okay, so over the last 50 years, for example, for CO2 methane separation, people have tested 484 polymers for CO2, 414 polymers for methane. That's everything we know. Okay, so we know 10,000 polymers exist. Of them, people have tested only about 400 polymers. Why haven't they tested more? It's a very expensive process to do that, because you have to conduct the experiment, varying pressures, make sure that you're doing it right. And so over 50 years, you're getting that many data points. If I was to test 10,000 more data points, it's going to take till you guys are old, whatever. Okay, so time is working against us. So I'd like to know which polymers are going to be better so that I can start going up here, high permeability, high selectivity. That's the goal I want. So, why do you get ahead of me? That's I'm coming to. So I'm going to talk about machine learning, right? And so the idea is that the total number of data points is 600. Yeah. And I'm going to use some to train the machine learning algorithm and some to test. That's always going to be 80-20 split. So that's where those things go. Yeah. Any other questions? Anybody else want to give the answer away before I get there? No. So basically you can see lines here, right? So the idea is that in 1998, when they drew the line, all the data points fell below that line. In 2005, it moved up here. In 2021, it's moved up there. So there's no understanding whatsoever. It's a line that bounds all the data below. So it's completely empirical. There's no understanding of that line. And those are sort of the questions we want to go after. Okay? There's another problem here. You see, there's 300 or 500 or 600 data points here, right? I don't know which polymer corresponds to what. So I have no idea, given it, if I know a polymer structure, which one gives rise to high permeability, which has the higher sieve size. So I have no clue what the relationship it is between polymer structure and its sieving capability, or if you like, the size of the pore. So we don't understand anything about this problem. People have empirically gone in, shot the first 600 polymers through, gotten some data, started to get some correlations, and built companies that manufacture these membranes. Everybody following along. That's the state of the art. We think we can do better, right? Nobody following this train of thought. You're following along. So I want to get high permeability, high selectivity. I have 10,000 polymers that I know. Can I use information from these 600 to start figuring out if there's any of the polymers that are better? Okay, so that's the first dumb question I'm going to ask, which is completely practically motivated. Okay, and then I'll come back and say, what is it about those polymers with high permeability that gives rise to high flux? So I'm going to go back and try and understand the physics of separation later on. Okay, so my first step is going to be completely industrially driven. I'm going to just ask myself, can I get better polymers? And then eventually what I want to do is the following, that I want to define a medium with high permeability and high selectivity, right? I want to get this definition, and I want to go back and figure out which polymer will correspond to this permeability and selectivity, okay? That's the overall goal. Right now, what people do is the way we are all taught to think is given a structure, can I predict permeability and selectivity? What I really want to do is go backward. So that's the inverse design problem that I'm after, okay? So what I'm going to try and convince you of is that this inverse design problem needs us to really understand the relationship between structure and permeability, so-called the forward problem. If I get the forward problem sorted out, then the inverse problem follows up. So I really want to design materials, which I think is one of the titles of this workshop. That's one of the things I want to do, right? So I want to design a material with the known permeability and selectivity. But to do that, I need to know, given a structure, what the permeability and selectivity is. All right. So this forward problem turns out to be the bottleneck that we're going to keep on trying to solve. All right. And I'm going to take a case where the forward problem is known, the forward answer is known, and tell you how the inverse design is going to work. But for this problem that I'm after, I don't know the forward problem. I don't know, given a polymer structure, whether it's going to fall here, there, or there. So I really want to start understanding how to create forward problems for these problems with unknown physics. You can ask as many as you like. Go ahead. I can hear him. He doesn't need a microphone, but they do. Okay, go ahead.
As I think, machine learning is something like the experience. Like we grow old, we see things, we learn. So is it that a fair statement?
That's an interesting question, right? In the following way, what you're telling me in life, experience is basically a correlation.
Yeah.
Right? If you see a car coming at you, it's going to hit you, it's going to hurt. That's correlated, right? So that's the way machine learning is used right now.
Yeah.
Right? But I'd like to go beyond that. I'd like to actually understand what is it about the structure that gives me this permeability, and then it goes beyond experience.
Yeah. So let's say there is, so out of these 500, 600 polymers that the some company make, so the person who is responsible, who is thinking of, okay, let's see, that polymer might work. So in that, coming up with next polymer that person is thinking of, there must be some learning or must be some experience which is working. And if somebody asks him, why do you think that's next to your proposal, that X polymer is going to work? There is something understanding already there with that person, right?
Yep. So that I didn't go into this. So look at these red data points. Those are 1998 points. These are between 1998 and 2020.
The blue ones.
Blue points, right? So you can see the points are going to higher permeability, higher selectivity. So in line with what you're saying, what they realize is if they make the polymers stiffer, then they pack worse. If they pack worse, you have higher free volume. They are more permeable systems, right? That explains higher permeability, but it doesn't explain higher selectivity, right? So the intuition goes so far, but no more. But again, what people have tried is a small subset of the polymer. So you wake up in the morning, you decide, I want to try polymer X because I had a dream about polymer X, right? But that's only your dream. There might be reality somewhere else that's better. And I want to stop dreaming. I want to do something that might have something to do with reality.
Yep. Okay. Thank you.
Anything else? Use the microphone, dude. Otherwise, they'll come and hit me.
Yeah. I think all the data points are experimental data, whatever you're showing in this chart. So you know exactly corresponding polymer. What are this depending upon the selectivity. Now you are kind of using, for a machine learning purpose, you are kind of back calculating the things. That given the selectivity, I just wanted to note what kind of polymer is it. So, but that is not a single parameter, because why this is the selectivity, there is not only single parameter, there might be multiple parameter, a lot of chemistry involved also.
Absolutely.
So then beyond the selecting that particular polymer, we can't get any other information.
So, you're saying something which is really important, which is here, I only talk about polymer structure and properties. Right?
Yeah. Yeah.
Now you might come up with, suppose I pick this point, I might pick any one of those 4 or 5 polymers equally likely, right? So there's a uniqueness problem in doing the inverse problem. Inverse problems are non-unique. We know that. Okay. But now you come in and you say these 5 polymers exist. Now, as somebody who wants to make money, you say which is cheaper, which is easier to make, which is not going to harm the environment, which is not going to poison somebody. You start throwing additional constraints to refine your problem. So at the moment, the problem is underdefined because I'm only looking at it as an engineer who wants to make a better material. But when you go to a product, you start adding additional things, which will narrow the choice. Now, there's another choice that's very important. It's called synthesizability. So I can make polymers up randomly, but that doesn't mean you can make it in the lab. So again, these are constraints that you can start adding to make the problem more complicated or more realistic, if you like. But I'm illustrating by taking the simplest problem that's out there. That fine? Anybody else? Pretty good. People are awake. Okay. So I'm going to sort of illustrate this forward problem and tell you how it's so very important by picking an example where there is a forward problem, or at least there's an approximate forward problem. So the idea is the following. I'm not going to do this. I'll come back. I think my computer sort of died, which is good, right? So, people have been doing this system of assembly of DNA grafted particles. So the idea is the following. You take particle type A, which has single-stranded DNA on it. And this is Paul Alivasaros and Oleg Gang and several other people. You take particle of type B, which has single-stranded DNA. These are complementary single-stranded DNA particles. You go below the DNA-based pairing temperature, these things start pairing up. There's an attractive interaction between the particles. And now, depending on how many of these you have, because all the pairing needs to be done, every A will have a certain number of B neighbors. And so therefore, by picking the size of the particles and the number of drafted DNA chains, you can pick out a morphology, okay, that the thing will crystallize into. Everybody satisfied with what I just said? Or have I lost you? You following along? Okay. So it turns out that people start thinking about the number of nearest neighbors, particles, the number of the size of the particle, the number of graphs, and they came up with some forward model that says, I calculate this parameter, whatever it is, and depending on the value of that parameter, I get a certain crystal that'll. Okay, so the idea is that the ratios of the sizes of the nanoparticles and the number of linkers are the two variables that are found to control what crystal lattice it goes into. So this is now from a paper by Chad Merkin and coworkers. They found that depending on size ratio and linker ratio, they got 4 kinds of crystals. Cesium chloride, Cr3Si, which means in this particular case, that's a salt-like crystal. A and B particles alternated into a simple cubic lattice and so on and so forth. They found these 4 different lattices. So what we asked ourselves first is, given that they can predict these 4 different crystal sizes or crystal types based on this one parameter, which is some sort of a forward model, are there other crystal structures that are possible for these systems? Experimentally, they found those 4. We said, can the other structures, if you go look in the database, there's something like 500 known crystal morphologies, right? And so we said, can we pick other size ratios where we get other kinds of crystals? So how do you do that? You pick your favorite kind of crystal type. Let's say PD5PH3, right? That's a particular crystal structure that you want to form. And you ask, what is the size ratio and the linker ratio that would give me PD5PH3? How would you do that? That's an optimization problem. Okay, so what you do is you pick a population of the 2 kinds of particles. You take 100 different kinds, right? That's the starting point. By using the forward model, for each one, you predict what crystal structure it would form, okay? It may or may not find the PD5TH3. If it doesn't, there's a distance function. There's a penalty if you don't get the right structure. And then you take the ones that are closest and you genetically modify. This is called a genetic algorithm. Basically, what you do is you generate 100 or whatever number of initial starting states. 2 of these might be really close to the answer. The others may not be. In the next generation, you keep those 2. And then what you do, the others, you start doing some sort of mutation. That means you change the size ratio, you change the ratio of the linkers. This is just a game you play. And you keep looking as a function of time whether you get the right answer by going through and many, many times implementing the forward model. Have I lost you guys yet? Or everybody following along? It's just an iterative way of optimizing and finding a solution. That works really nicely. So as long as you know the forward model, for every size ratio and linker ratio, you can predict what crystal forms. And so we've gone back now and found that in this part of the phase diagram, you get different kinds of crystal polymorphs beyond what was seen before. What I'm trying to tell you here is that if I know a good forward model that tells me how I can correlate structure to properties, Then there are optimization algorithms that exist that can go locate a particular structure. So if I have a good forward model, I can do the design without a problem. There are established tools. I really don't care about that. Okay. So I'm going to sort of make a case that I want to do materials design. The tools to do the design exist, but the forward model going from structure to properties is what's missing. That's what I have to come up with. Everybody satisfied? So really what I want to focus on today, how am I doing for time? 4:30, right? That's the right time. All right. So I told you that if I have an analytical model, if I know the connection between structure and property, I'm done, right? But for the gas separation problem, I have no idea what the relationship between polymer structure and permeability or selectivity. So we need to find models from the available data. That's what I want to talk about today. And then it turns out as you go further along, you have so little data that you have to start thinking about how to get the data that is the best way of building a model. That's called design of experiment. So those are the ways of building forward models. Today's lecture is going to be completely focused on how I'm going to do that. How am I going to generate a model when I have no understanding of the physical situation? Is it late in the day? Is it boring? Or is it trivially obvious? Or all three of the above? How are we doing? I'm sorry, what? Optimum, really? Okay, that's the first time I heard somebody say that about my talk. Most of them are like, oh. All right, so I've already defined the problem, right? Here's 500 polymers, roughly, that people have built, they have experimental data for. I know the structure of each of these polymers. I want to understand a model between structure and permeability. And then I want to see whether of the other 10,000 polymers that have not been tested, are there ones that are better? That's the question I'm going to ask. And of course, later on, I want to understand the physics of the separation. That's what you guys care about. We'll get to that later on, okay? I'm going to just think about how do I get this better polymer so that I can go sell the stuff, get money, and then do what I really want. Okay. So polymers, is basically monomers connected together to form a chain, right? The basic building block is this monomer, which keeps on getting repeated, okay? So I'm going to start by saying, can I take this monomer and establish the relationship between monomer and the permeability? There's also other variables like the chain length, but I'm going to argue for the moment, and there's evidence to suggest that that's okay, as long as the chains are long enough. The chain length really doesn't matter. It's just the building block that matters. So I have to correlate this guy to permeability, okay? So how do I do that? I need to go from here to there through some sort of regression. And to do that, I need to understand how to represent this molecule to a computer. That turns out to be a really difficult problem, okay? Because I need to do two things. I need to be able to maintain the structure completely, describe it completely to the computer, all right? But I need to do it in a manner that is unique, but also not have so much information that it's going to blow up the computer, okay? So let's imagine here, if I think about this, there's carbons connected to carbons. You guys know enough about that chemistry, right? As physicists, nobody has problems understanding that that's a benzene ring, for example, right? So what you start to do is you fingerprint this molecule. That tells you locally a benzene ring is attached to a carbon here, to another carbon here, carbons connected to an oxygen. That's at the nearest neighbor level of structure. But I want to understand how the whole molecule is put together, right? So I'm going to have to put in not only local structure, but non-local structure as well. So that's what makes this the string that describes this molecule more and more complicated, okay? And the problem is that if you try and make it really unique, that means everything is going to have one particular fingerprint, and there'll be nothing shared across the different molecules. I have to have enough sharing so that I can say, every time I have a benzene ring, that's going to give me some contribution to permeability, but also this molecule is different from other molecules. Am I doing okay? You're following along? You good? You seem happy, you're smiling along. No? So this idea of fingerprinting is something that is very well known, and what it ends up telling you is you have these numbers of zeros and ones, and typically this fingerprint for a monomer is going to be of the order of 5,000 bits, okay? So it's a very large amount of information that is stored, and so I'm going to have 5,000 bits per monomer, and I have 500 polymers. So this is now starting to add up in terms of data. There's a lot of information about these 500 different polymers. And what I want to do is correlate the fingerprint to the permeability. Yes.
Curious, why did you fix these two requirements for fingerprinting, fixed length vector and group contribution?
Oh, because the idea is that, you know, at the end of the day, if I have a non-fixed vector, then imagine some molecules are going to be short, some are going to be long, and I'm going to have to fill it up with zeros at the end. So I pick up the largest molecule that defines the longest vector and then define everything on the basis of that longest vector. It's just easier to do the math.
Size vector you get through a hash function.
We'll talk about hashing later on. The reason hashing is a problem is, everybody know what hashing is? Okay, hashing is the following. You go to a Mac machine, you enter your PIN. Okay, when you do online in India, you see that. You enter your password, it immediately replaces it by some very long string, right? What that is doing is it's hashing it. You enter your password, it gets hashed into something which then is compared to what is stored on the bank. Okay, so when you go from your password to the hash, it's a one-way. But then if I was a thief, I stole the hashed one, I should not be able to reverse it back. Following along? So that means many molecules will hash into the same thing, because that's how I don't get a non-unique solution, which means going back from the hash to understanding stuff becomes problematic. That's why you don't want to hash. We all do hashing, it's a problem. Everybody else didn't understand it, don't worry about it, take a nap, we can come back.
You want a one-to-one and onto map.
I want to have a one-to-one if I really want to have something interpretable, right? If it's, and hash algorithms are set up to not be one-to-one, that'll create a problem. There's a lot of bit collisions that it creates. Narayan.
You have to include information beyond chemistry along the chain. Do you have to say how crystalline this thing is, or persistence lengths, this kind of thing?
I mean, it turns out from what we find, if I tell you this is the monomer, that seems to be sufficient. Zero thought. I mean, that's something shocking already. I mean, I had the same question. How much information do I have to give it? I have to tell it how long the chains are, what the phase of the moon is it crystallized? Are you sneezing when you do it? All that information is what I started thinking about. But it turns out all I have to tell you is the building block monomer structure. That was not at all obvious. That's telling you something really powerful. I don't know what it means yet.
I see. So for the same monomer processed in different ways, it seems to have the same pi alpha.
It seems that whatever variations that are caused seem to be smaller than the standard deviation of the data you have.
I can make the stuff in any way I like.
Yeah, so it doesn't really matter. But I don't know the answer for why.
Just like this following kind of questions, because here you are only bounded with 0 and 1. Because by 0 and 1 only we're defining your monomers one chain. But there are a lot of like carbon, oxygen, nitrogen. If you keep on increasing, different kind of monomer might have different kind of elements also, and different bond angle, just like sir was telling.
No, don't go to bond angle. All I need to know is what is connected to what.
And still that is also 0 and 1 is not enough, I think.
I think it's perfectly enough. If I'm sitting next to you, it doesn't matter if you're sitting this way or that. That's built in. If there's a carbon, nitrogen, carbon bond.
But let's say now introduce oxygen in between. Now it is not carbon, nitrogen, carbon. Now carbon, nitrogen, oxygen, let's say.
So those things have to have different bits to distinguish those two, right? So if I have a carbon, nitrogen, oxygen, carbon, that will have a bit here, which will be 1. Whereas a carbon, nitrogen, carbon will have that bit 0, but 1 somewhere else. So I'm going to use just the connectivity as a proxy for all the geometry in the molecule. So you're thinking like a chemist. I'm saying, don't do that. Think like a data person for whom everything is zeros and ones. Is that okay? Yes.
Yeah. I think question as raised by raised there.
Yeah, Narayana.
Yeah. I think the point of this, whether what kind of crystal and so on, that goes already into the problem while you train the system. Because for the training, you are using already some polymers, which those polymers already have the information regarding crystal structure and so on.
Sure.
And when you train, that information is going in.
So I'll give you the counter to that. If you think about polyethylene or polyethylene oxide or any of these semicrystalline polymers, depending on how you process, you can get extremely difficult degrees, extremely different degrees of crystallinity. All right. So encoding just polyethylene does not tell me the amount of crystallinity. Okay. So depending on how I process, the crystallinity varies. The fact that it doesn't matter tells me what matters is the chemical structure, not the crystallinity. Okay. I mean, that's not a conclusion that's obvious at all, but that's what this is teaching. So we have to go back and understand that. I have no clue at the moment. I'm just giving that as a piece of information that somebody smarter than me, like Kumar, will come and figure out. Yeah.
An upper limit on the length of the vector, you're kind of putting an upper bound on the complexity of the constituent molecules, right?
Yeah.
How do you choose that upper limit? Is it just based on a really complex thing that you think nothing would be more complex than this, or just?
It turns out that typically people have picked 2048 bits. It turns out nothing has turned out to be bigger than 2048 bits. Okay. Now, the reason for that is sort of obvious. People who go and work in the lab are going to take simple molecules to test. So the number of things you're going to have small. Now, as you want to expand out, complexity is going to get larger. So then the bit size gets bigger. Then going back to the question he raised, is that molecule synthesizable? Is it pragmatic? So I suspect as you increase complexity, you get more interesting phenomena, but less commercial relevance. That's not something we're trained to think about, but that may be the compromise. Yeah. Yeah.
So do you think about, is there like an optimal way to like represent certain things? Like, for example, you have this benzene. Do you want to have a separate signature for it, or do you want to think about like carbon, oxygen, all the things individually that would make,
No,
you can make everything, but then that would require more
Right.
space to put.
And that's always the balance, right? I mean, in principle, if I can straighten this thing out and have a linear sequence that I can map, that's the most convenient way of doing it, right? But it's going to be very expensive, right? On the other hand, if I take a benzene represented by one unit, which we all agree is benzene every time, that makes life easier. So this idea of fidelity versus cost is what drives the decision. There is no answer. You play, and when the answer falls apart, you say, I made it too simple. Let me go back and make it a little bit more complex. It's all trial and error. Okay? Answer should be independent tokenization. Yes, absolutely. But you don't know that. I mean, you want to start as simple as possible. It might give you garbage, and then you make it progressively more complex where it stops you. But there's no rule, there's no rules to do that. It's all empiricism. Yes.
Yeah, I was curious, have you thought of also using AI to do the fingerprinting?
I'm sorry, what? AI too?
You considered using AI itself to do the fingerprinting?
You mean like a neural net or something like that? The problem is, think about where neural nets work when you have tens of thousands of data points. Okay?
You have thousands of polymers, and you could also feed in a lot of other parameters apart from the structure.
Fair enough.
The energies and so on. Maybe it does the fingerprinting in a high dimensional space, which could be.
I mean, the point's very fair, right? But I'm thinking like an idiot. I want to start as simple as possible and make it no more complicated than I have to. So I'm starting at the simplest level, smallest number of polymers, working my way up, and see how far I need to go before I can get away with it.
So minor thing. So is this done by hand more or less?
So there are now programs that exist that will take a structure, you put it in, it'll actually convert it to a string like that, right? But before that, it had to be done by hand. Now there are converters that will actually do that for you. But it's the same idea. It's still very, a person sits there and hacks their way through. And the way we do that is we hire undergraduate students and we buy them pizza and they'll do anything for pizza.
So, simply put, what you're doing is applied information theory.
Yeah, absolutely.
you have 108 elements, so you have an alphabet.
Absolutely.
And you're looking at the most frequent alphabets.
Absolutely.
And you're giving them higher
Absolutely.
values. And from there, you're taking the patterns, which turn into your words. That's the simplest way to go about it.
That's exactly what we're doing. Nothing more. It's old information theory that we're using. Yeah, exactly. Yeah. I mean, this the reason, of course, you can see why I'm doing it. Established technology, it's hard for me to screw it up. I'm going to take a detour, right? When people talk about data science, they're talking about big data. How many of you have Amazon accounts? A lot of people. When you watch a movie, what does it tell you? Based on your experience, you're going to watch this movie, right? That's because there's 6 billion people living on Earth, each of whom has seen 100 movies. So it's 6 times the 10 to the 11 data points that they have to work with. That's large data. This is 500 data points. Is this large data? Is it small data? How do you start to decide large versus small? That's the first question I want to bring up. So I'm going to take a detour to tell you where these methods might start to even apply. Where is it appropriate? I think you brought up a question of neural nets and stuff. Where is it appropriate to start thinking about the data tools? When do I start thinking I have enough data to do a neural net versus machine learning, right? So this is something we've thought about a lot. And I'm going to walk you through this. So you take that string. Remember this string I created? You can think about this as an array, right? Where if it's 2048 bits, every molecule is defined by 2048 bits. There's 500 polymers that I know, so I have a matrix of 500 by 2048, all right? I construct what's called a covariance matrix. How many of you know what that is? I'm seeing puzzle looks. A covariance matrix is this. The variance, sigma squared, of a one-dimensional data set is summation xi minus x bar squared summation over I 1 over n. That's the variance, okay? The covariance, for example, of xy all right? So it's just basically like you calculate, this is a second moment about the mean, but it's all the cross moments, all right? Everybody following along. So this covariance matrix tells you all the correlations you have across all the bits and all the different polymers. Okay, so that's the covariance matrix, all right? I'm going to take the covariance matrix, I'm going to find its eigenvalues and eigenvectors. Everybody understand what I just said? The eigenvalue tells you which is the direction. So the eigenvector corresponding to the largest eigenvalue tells you which direction has the largest variance. Okay, so you order them in order of decreasing eigenvalues, and you then start counting up the eigenvalues divided by the sum of all the eigenvalues tells you what fraction of the eigenvalue came from that principal component. Do I have to write all that down? You got all that. Got it. Okay. So you start asking yourself, if I have this 1024 by 500 matrix, how many data points do I really need before I calculate all the variance? So that means I have a chemical structure made up of this monomer. How many monomers do I have to consider before I capture all the variations? So that means everything has carbon, everything has oxygen, everything has hydrogen, but they're connected in different ways. And I'm asking, how many monomers do you think I need before I've covered all the variations that you see in all the different molecules? That's a question. Anybody have a clue? At least did the question make sense to everybody? There's 500 molecules I've tested, right? Each of them has a 2048 bit. They're all different kinds of monomer structures. How many monomers do you think I have to start telling you information about before you've seen all the groups, is what I'm asking. Any idea? The question makes sense, right? You follow the question or you lost? Okay. So this is very important question because this tells you how many monomers do I really need to see before I've represented everything in the database, right? And this number, I did not know what to expect. So I'm plotting here the cumulative explained variance as a function of how many eigenvalues I maintain or how many principal components I maintain. So this general figure of merit is you need to get to at least 99%. before you've explained all the variations. And that turns out to be about 300 points. That means I need to tell you at least 300 monomers before I've captured all the variance in the monomer structure. Now, in this case, it turns out people have tested between 300 and 500 polymers. So they just barely have enough data to capture all the variations in chemical structure. So we are really working at the low end of what is admissible for data science methods. So these are, everything we do here are called small data problems. Small sounds pejorative, so we call them precious data. Okay, this is marketing, nothing more, right? But the point I want to tell you is that if I'm going to start using data science methods, but this kind of permeability data, I'm working at the low end of what I should be able to do. I have to be extremely cautious. Okay, and this is 50 years worth of data. I'm bringing this up because everybody who works in the physical sciences, for them, if they have 1,000 data points, that's a big deal. And I'm telling you, that's not a big data problem. That's very small data. And so you have to be very cautious what you do. Ranjini? She couldn't take it, she's leaving. All right, so now I'm going to actually tell you what we did. I've taken a long time to get here, right? So I need, the formulation is the following. So I'm going to come back to my permeability problem. The X vector is that chemical fingerprint. That's that 2048 bit. The Y is a scalar, that's the permeability. This is called kernel ridge regression. It's whatever, it's one of the machine learning tools that's out there. It's basically trying to minimize this function, the cost function, which is the relationship between the chemical fingerprint and the permeability. And the postulate is that they are connected through a linear transformation, which is this matrix W transformation. That's all machine learning is. It's a linear transformation, one space into the other. So the point you want to make here is I want to minimize the square because that's minimization of the error. And I want to determine the elements of W transformation. How you do that is standard optimization. And then there's something they do, which is called regularization. So this here is the norm of that W matrix. And you include that in because you want to make sure that the elements of W are as close to zero as possible. So you're not dealing with large positive and large negative values. So this regularization trick keeps things as small as possible, the elements of W. That's the problem. You want to minimize C, you want to find the W that minimizes C with the constraint that W's elements are as small as possible. Nobody following along? This is a very simple optimization. You can go through the math and you end up with that. The W equals that. That's just taking it, doing a first derivative and solving that. That's all machine learning is doing for you. It's solving that transformation and gives you elements of W. Now, how do you do this? How do you fix it, right? How do you make sure it's doing what you want? So the idea is you take your data set. So I have 600 data points, for example. You break it down in some manner. Say you break things down into what's called a training set and a test set, which means something like you pick 80%, say, at random of your polymers. You train it. That means you calculate W for that 80%. And then you test it on the 120 that have not been used to generate W. And if you did this properly, if there's enough data, the training set should give you good estimates for the test set. Okay? Everybody satisfied? Now, what we can play with here is how big the training set should be. And I've already told you that if I have less than 400 points, I'm not capturing all the variance in the chemical structure of the polymer. So it should tell you that if the training set gets smaller than 300, my error should be large. And if I get larger than 300, the error should start dropping. Okay? So let me check that. So here is the mean average error in my permeability, plotted as a function of the training set size. Let's pick your favorite one. Let's pick methane. So this is what the model is trained on. Okay, so this tells you it doesn't matter how big a data set I use to train it. Once I fit W to the data, it does a good job reproducing the numbers I gave it, which is sort of dumb, but you make sure that that's okay. All right, so this is just telling you I fit W properly. Then I use the W I generated to go back and look at the remaining points on which the model was not trained. So if that number is less than roughly 300, you see the error is of order 1. So it's telling you If the training set's smaller than this 300 or 400 data points, I'm not getting anything predictable because I haven't captured the diversity of my monomer structure. And then once I make the training set larger, the error starts to fall off. But you can see here, even for the largest training set I use, the trend is still going down. I just simply don't have enough data to have the lowest possible error estimate. Again, emphasizing to you, people will come and tell you use machine learning, use it, but use it with extreme caution because the kind of data sets we all deal with are really, really, really small. Okay? So it tells you with all of these polymers, you need of order 200, 300, 400 points before you start the error to drop. And then you should definitely have much, much more data before I would really start to test. But what is a good metric where this is really working is where the test set and the training set have the same sort of error. That means they all have enough information that I've captured all the variation in the training set to go into the test. Narayanan, do you need a microphone or are you just going to scream? I know they won't like you for that.
So what you want as an outcome is to actually pick the very best ones, the outliers. So in the orange, in the whatever that color is, are the big errors coming from things that are, you don't care if you get the typical ones right as much as being able to get the outliers. Are the errors coming from the outliers in P and alpha, or are the errors coming from, are you able to catch the best ones as well as?
I don't know the answer to that because what you do in these cases is you don't just do one training set, right? You fix the training set size, you randomly pick different parts of the data, train it, and you average the W from different training sets and use that on the test set. So I don't think you try to distinguish outliers versus median. I just think in an average way, this is doing badly. So I'm not asked the outlier question. So I don't know the answer to that question. Right? But I think here, the fact that it's flat basically tells you I've not captured the diversity of the monomers. So I'm definitely going to be screwing up because I'm not going to, when I go to a new polymer, there's going to be something in there I've not seen before. I can't make a prediction. So the error is going to be 100%. That makes sense? So now this is how well it can do after you go down to the, when you have 80% of the data, that means you're training it about 500 data points or so. This is the predicted probability versus the actual permeability. Pick your favorite gas, helium, for example. The blue is the training set, the yellow is the test set. And you can see some outliers here where it's starting to fail. But in answer to your question, Narayanan, it's not really happening at either edge of the permeability. So it's somewhere in the middle, just random stuff going. So you can do pretty well. So this tells you I have a model that's properly trained, right? Now what do I do with this thing? So I trained it on 500 data points. Now I go back, there's a database that has all 10,000 known polymer structures tabulated. It's somewhere in Australia. We went and downloaded all the structures. And right after that, they created a firewall. None of you guys can go in there because now you have to pay money to get it because they figured out this stuff is useful, all right? But here is 10,000 polymers. Let's just look at oxygen-nitrogen separation. It doesn't matter, right? This is permeability of oxygen, selectivity of oxygen to nitrogen. This is the current best polymer. That's the current upper bound. These are all the polymers that have ever been made. And you look at it, everything follows below that upper bound. So it tells you people who were picking polymers for this particular separation in line with what you said, their intuition was very good. You're not able to do better than that for this separation, okay? 1 point for you, 0 for me, right? On the other hand, look at CO2 and methane, or look at this one, CO2 methane. Here, there's a very large number of points that your intuition didn't work. Okay, and this is the separation I really care about because this is a greenhouse gas that I want to get out. So there is use in intuition, but it doesn't go all the way, right? Somebody have a question or is just people wandering around? So this is great, right? Because it tells you for CO2 methane, I'm able to predict things that are better than anything that's ever been tested. Now, this better not be a figment of your imagination. You really need to go and validate whether that's right. How am I going to do that? Yep.
How was the upper bound established?
This is the one that I showed you in the first slide. That's the current experimental best polymer line.
So when you say that that's the upper bound, so for oxygen nitrogen separation, you can't do better than that. So even the upper bound was arrived at through empirical.
Correct. So what they did 500 polymers.
There may be polymers that fall outside that upper bound and the upper bound will move again.
No. Okay, hold on. So that's my point. So the current people tested 500 polymers. Based on that, they drew this upper bound line. I now know there are 9,500 other polymers. I test 9,500 polymers. None of them go pretty much above the upper bound. So I said, in that case, those guys got it right. We could ridicule people for empiricism. They got it right for this one. So the upper bound is not going to move if you go and test another 10,000 polymers. Whereas for CO2 methane, there clearly are many, many points. So I don't understand why these two separations are so different, but apparently people who studied this were smarter than the people who studied. That's the only thing I can do. So now it turns out these are the polymers. These are just two polymers that are predicted to be better than the upper bound. I have a synthetic colleague who said, oh yeah, I know how to make that. He made it. We tested it. This is the prediction that the machine learning gave us along with error bars. The red point is the experimental data. Is it perfect? No, but it's in the right direction. So here the machine learning is validated by experiments, and clearly now we have a polymer that's better than anything that's been tested. Question now is this in terms of value something that you can make, something that you can use? And that's the question that we have to ask. That's what we're doing right now. Okay. So here is a success where small amount of data is used to make polymers that are better than anything that exists for gas separation. How am I doing for time? I can keep going. Should I stop and let these guys do examples? I mean, it's late in the day, right? Is my student on? Can you check? Give me a second. You know, the problem is that it's 6:30 in the morning for my student. He might be asleep. Let me just check. Where is the Zoom window?
I'm in here. If you can,
Okay.
hear me,
They just, you want to take it away? You want to, is this too much time for you or you got it?
I can work with anytime you want. So if you want to keep going, it's all fine. But you know,
I'm
up to you.
done here. So this discombobulated voice is my student, Tejas.
Sure.
He's amused by my joke. That's why I pay him his salary every month, right? So he's actually put together, Tejas, you want to talk to them about what you're doing and then how you're going to do this thing?
Sure.
Go ahead.
So I don't know if I'm still a ghostly voice, just speaking from the ether, but hi everyone. So my name is Tejas. I'm a third year, fourth year, somewhere in that range in Dr. Kumar's group. And so we have a handful of demos for you that are going to go with all these lectures, and can sort of jump right into any of them. I was told that everyone was loosely comfortable with Anaconda. I can't necessarily see anyone, but if there's anyone that is not comfortable, speak now or forever hold your peace. And I'll trust Dr. Kumar to tell me if anyone is raising their hand.
Anybody not have Anaconda on your computer? You guys want to fire up your computers to see what he's doing along with him? Or you don't have to. You're going to walk them through, right, Tejas?
I can. They can step along through either way.
Why don't you go ahead and start, and then maybe if people want to ask you questions, they can. But share screen and we'll go from there.
Sure. Okay, I'm gonna boot you now. So, yeah, here's just Anaconda Navigator. So I'm gonna do this in a way that if anyone wants to follow along, you're more than welcome. So sort of give people a few seconds to launch the navigator. But from here, you're gonna go to just the Jupyter interface. And this will sort of bring up something in your preferred browser.
I didn't do any of the design of experiment. I've only done the machine learning part of it. So I've only done so just go with that and don't go into design of experiment, just do the first part.
Yeah, no, that's probably day 3.
Thanks.
Okay. So wherever you unzip the files to, just navigate over there in this menu. And you're looking for membrane regression for anyone who's trying to do this live. So go in here and we'll launch this. I'll give people a few seconds to sort of scan things through, but the short of this here is that the model predictions that you were just looking at, this is going to step you through how we did it. So there's a little bit of a preamble here about how we're using the database, and I can go ahead and pull that up. So if you look at the actual file that it's calling up here, it's this perm data. And this is just a massive spreadsheet that has the polymer's name, the smile string, permeability and bearer for each of the gases of interest, and temperature and pressure from which the experiment was performed. So keep this format in mind. And we'll go back here. So there's some extra reading material here if anyone wants to dive back into this later. We're going to start by, as always, importing all of our modules. So we'll run this. Anyone who's following along, I think you may not have this, the RD kit, but if you don't, you can just go down here and you can do a pip install RD kit, and that should take care of anything for you. They just added this functionality a couple months ago, so you no longer have to build this from scratch. Obviously, mine's already installed, so.
You want to talk about what RDKit does and what the max keys are?
Yeah, so RDKit is just sort of a general chem informatics Python package. So it has tools to sort of get the computer on the same page as you. It'll give you an interface to work with molecules and to break down different parts of them. So you'll see there's a part in this this notebook here that's going to go through how to print each atom in a molecule and get atomic numbers and do stuff like that. You can do similarity searches. So if you have two molecules in Python and you want to see what's shared between them, RDKit gives you a very clean, easy way to do that. And then it also gives you featureization techniques. So you have different ways to fingerprint molecules. There's the standard RDKit topological fingerprint, which I believe is what you've been looking at the whole time. And then there's something also called max keys, which is what we're working on a separate paper right now. Max keys is slightly different in that it just sort of looks through predefined motifs for each index, and the values are still binary. 0 for this motif is not in the molecule, 1 for it is. So hopefully anyone who wanted to install this has gotten their installation through. What we're going to do here is just call the perm data Excel spreadsheet here. So we're using the pandas module. So we have PD because we imported it as PD. Sorry, again, if anyone is super comfortable in Python, just bear with me. I'm gonna go very slowly. So we have pandas. We're gonna read Excel, and you're gonna get a whole bunch of messy stuff for some of the rows that don't necessarily have data in them. So we have, column 0 is going to be the name of the polymer. Column 1 is going to be smile strings. We have all of our permeability values, temperature, pressure, and then we have all these garbage rows and columns that don't necessarily serve any purpose. So we're going to split them here. And what we're doing is just essentially taking the polymer, the smile strings, and then the permeability values for each gas, and we're making separate data sets. So I'll go ahead and run that. And then if you just do the head command in pandas, just gives you a sort of preview. And I've put 5 here, so that gives me the first 5 things in this data frame. And we still have to get rid of these not a number lines, but this is sort of just a trimmed down version. So CH4 underscore data is just all of our CH4 data. So I did initially plan on this being pretty interactive, but I guess we can just sort of chug through this unless people want to try and tackle this for a few minutes. I'll leave that to democracy.
I think you should just go ahead and do it, and then they can play with it later. That makes most
Fair enough. All right. So yeah, we have a helper function here called cleanDF, DF for data frame, that we're trying to define here. And it's just going to work on any object called df that you provided, which is just going to be our data frames. So I will drop in the cheat here. What we're trying to do is just get rid of the not a number rows, and Pandas has a very easy command where you can just do that, and that'll get rid of all of your not a number lines. And then on top of that, we want to drop any duplicate smiles rows in case we have two papers that are reporting the same polymer. We probably just want to take one. You could also think about averaging them, but we just wanted to play it safe here, so we'll just get rid of them. And the lovely thing here is that you can just do drop.
Hey, just hold on, there's a question. Go ahead.
Yep.
Can you zoom out? You don't have a computer? No, I understand. Can you zoom out a little bit, Tejas?
Zoom out to where?
People can't see it, I suspect, right? If you can make it a little bigger, that might be useful.
Oh, yeah, okay.
This is what happens with communication from 10,000 miles, you know, sometimes.
Yep.
Thanks.
So yeah, you want this line here, and then you want this line here. And then inside the drop duplicates here, the open close parentheses is just sort of calling the method. But on this one, we want to put an option on it. So we're going to say drop duplicates, but with a subset equal to, and then we want to say smiles. Right. So this is going to drop duplicates using our criterion to drop as the smiles column, which is what the square brackets and the quotation marks are for. So once we have this cell run here, we go ahead and just run this helper function on every single data frame. And then we'll do O2 data, and we'll look at the top 10 things in there just to check that this function worked. And it did not, which is great. Interesting. This is 90% of what I do on a daily basis. It shouldn't matter too much. So yeah, the sort of next step here, I'll just jump ahead. It's not really worth waiting on that. Next thing we're going to do is try and take the smile strings here and then just directly convert it to things that the computer can work with. So the way that we do that is in RDKit, you have what's called a mole object. And so RDKit is going to start using object-oriented programming, and it's going to take the smile string and just magically conjure all the atoms in the right order from what you tell it. So here, you probably just want to take like O2 data, see if anything else this probably. So in a data frame, when you want to call a specific index, you use .iloc, and this is going to expect coordinates within your data frame. So we want it's probably going to be, let's take row 2, and it's going to be column 1, right? Because counting starts from 0. So this will be .iloc 2, 1. And then that should run. And if you go ahead and just type mole right under that so that you print what you just declared. Right now, it's just a string. So what you want to do is, I'll do this all in one line. We're going to wrap this whole thing in parentheses. And from RDKit, we've imported this thing called the chem module. So we're going to do chem.mole from smiles. Hopefully everybody can still see the text. And then, yeah, now once you, I'll get rid of this. And as you can see, it just sort of does it. And then if you print them all, that you have a molecule now, and you can sort of read through each atom in here. So RDKit actually handles everything automatically. So it has hydrogens on here implicitly. It's not going to show you everything, but it knows what's going on. So here, just for, you know, simplicity's sake, we'll do it's and then we'll do right. So we're using atom as our, you know, implicit iterable, and we have a method on the mole object that's saying just get the atoms. And then for each one of these items that we get out of the atoms list, we're going to print the atomic number, which is also a built-in method on the result of this command. And you know, I have the printout already on there, which is minor proof, but you'll see if you run that, you get all the non-hydrogen atomic numbers. So this is where the actual confusing parts kick in. So we're going to start trying to fingerprint these so that we can build up our database for the actual machine learning. So we know that we need this to be fixed length, and we know that we want it to include as much detail as possible. So there's a link in here. If you want to click this, this is sort of like a rough and tumble breakdown on how all the actual fingerprinting works. It's way too detailed, and it doesn't really, you know, you don't need most of this. You could also just go to RDKit, getting started, I believe, is the Google. And then you have sort of user docs for RDKit. But in short, I have just provided you this function automatically because it's kind of a pain. So we have two functions here. One of them is for the topological fingerprint, which is what you've been seeing. And then one of them is for Max in case anyone wants to double back here and try this on Max in your own free time. So what's going on here is the first function here, if you just want to look at the fingerprint, this is, I'm just setting up an empty list so that I can put the fingerprint in something. This is a step that just solves a bug because for some reason it doesn't like different types of variables. And you'll see here I have the defaults in a comment. So this is the actual function here. You have the chem module. We're doing RDK fingerprint on the mole object that we've called up top. And we want to say the minimum size is 128. We don't want it to be too sparsely populated. You know, otherwise you'll have your input is just mostly zeros. So the default options on here, the minimum path is 1, the maximum is 7. And what this is telling you is how many bonds in a radius it's going from each atom in consideration. And then also we have FP size is 2048. So it's going to give you a 2048 length array each time you calculate a fingerprint. And then we're using 2 bits per hash just because, you know, make it as easy as possible to compare it to other people's in case we have to do any sort of bug fixing. And then we want to include agents because those can be pretty influential depending on the case. So once you actually run the function, it's just going to convert it to an array, and then it's going to swap it into this dummy array that I've set up here because otherwise it'll give you a whole bunch of issues with, you know, dictionaries or lists or all that stuff. Now, if you want to calculate this in max keys for your input, it's way easier because you just have max keys dot generate max keys on the mole, and then you send it to a bit string, and then you just turn it into a float. That's just to make sure that everything is the right kind of number when you actually go to use it. So we run this just to define our functions, and then we go down here, and this is just going to be a very quick test. So we'll say FP test equals calculate fingerprints. And then we'll use our null object from above. And if you want to see what that looks like, you get an array. Then we'll go here, we'll take out this line. And then we'll want to say print length of FP test. And you should get 2048. That's because I forgot there's an extra dimension. And there you go. So this is actually, it's giving you a 2048 by 1 array. So you have to just get rid of one of the dimensions, not the biggest deal. And then, yeah, if you wanted to do this in max, it would be I think it's get max. Okay. Same thing, and then we'll print length of test vector, and we get 167, which is exactly on par. If you guys want to look at the actual max keys, those are available in GitHub, nowhere else, unfortunately. So there's this page where they have the master key. So index 1 is presence of isotopes. There's an entire key for actinides, lanthanides, different columns on the periodic table. And then, some of these get into sort of structural. This is an oxygen or a sulfur between 3 oxygens. And, they can get pretty hyper-specific if they need to. So we'll start by just using our O2 data. And, word to the wise, whenever you use the copy method, put deep equals true, because Python tends to include things as a reference to other variables. So if you don't do this, and then you change ML data, it'll actually go back and change O2 data. So I've wasted plenty of hours by accidentally missing this option. So we have our data defined, and then from here, this is just going to be a very non-professional computer science way of making an array. So I'm going to start with the 0th one, and then I'm going to calculate a fingerprint, and then I'm going to stack the second one onto the first one, and then the third one onto the second one, and so on and so on. And hopefully this should, okay. So probably want to change this to 2 because I forgot to clean up the top there. That's also great. Okay, I am going to swap in this one. And I'm just gonna fly through this part. Okay, so this is going to create that array. It goes up to 671, and that's the length of that data set. So we are in the clear. Sorry about that. So now that we have our database in a proper array, it's going to be 671 by 2048. And that's going to be our X here. So we're trying to minimize the distance or the absolute gap between our data times the weights, which is just a model prediction, and then Y, which is the actual target values here, which is permeability. And we're going to minimize with respect to W, which is going to be the model weights. So we're changing the model weights in such a way that we get the model prediction as close to the actual values as possible, is the intuitive way to read this. We're leaving this regularization term blank here just for simplicity. So go ahead and run these. And this is just a double check. We get 672. We have an array of arrays. And then we go in here. We have this perms variable, which is storing our target values. It's just a convenient way to keep our Y values on hand. And you'll notice that some of these can be sort of all over the place. So what we want to do is we have to create our train test split. And in this line, I'm going to create a train test split on FP, which is storing all the fingerprints. But instead of just directly accessing perms, I'm going to get base 10 logarithm of perms. And this is just sort of standardize the playing field here. We don't want something just absolutely blowing out the scale and having the model only pay attention to permeabilities above 1,000 or, a similar situation like that. And scikit-learn is very easy to deal with. So you just have this parameter train size here. And we're setting it to 0.8 because we want 80% of our database to go into the train state. And then this random state option here, just set it equal to your favorite number. And this is just common practice. So this train test split is going to be a random subset, as Dr. Kumar was talking about. So when you set random state to the same number, it makes it reproducible. So you never lose the seed of the random number that you actually use to go for it. So we have our sets, ignore this line. And this is just going to be a very silly example with linear regression, because why not? This is usually what we try first. So I have a note here that all the models have the same flow. I don't know if anyone's super familiar with scikit-learn, but basically all of their models, you import it, you instantiate the model, you train it, and then you get your performance metrics. So you can pretty much just swap out lines at will. So we already took care of this, the base 10 logarithm here. So we have linreg is going to be a linear regression model, and then I'll zoom in again if I can. And yeah, so we have a linear regression model, open close parentheses, just to tell the Python that, make this model. And we're going to do dot fit on X train and Y train. So you'll see that it prints, you have a box here. Now it tells you that your model has been created. And you have a bunch of different options on it, and I can actually show you in VS Code. So if you type in linreg, which is your model, you dot, this is a list of options that come up. And these are all things that are sort of built into scikit-learn that you can just call in one line if you need to, check things. Since this is linear regression, we have intercept and, things of that nature. So we don't actually need this cell or that one.
Shall we stop maybe, because I think people are just watching you do it. I'm going to
Up to you.
Yep. I'm just going to suggest you guys that everybody has this software, right? Run it if you like. Tejas is available to answer questions. I was hoping this would be more interactive. It's clearly not, right? So I'm going to let this be as something you guys work on if you like. But there's a couple of things I want to do. One was I want to show you how using a Python script you can actually do it. All our data is available, so there's nothing hidden. And you guys can go and look and figure it out. There's other pieces of software that Tejas has generated that's part of that file that he sent out. You guys can run it on your own time. If you need to ask Tejas, he's available. I'll give you his email later on. But at the moment, I think we'll stop. And next couple of lectures, we will not have a demo. We'll just do 2 straight lectures. All right. Thank you so much. Thanks, Tejas.